Hello, my name is Maya Shoemaker, and today we'll be discussing hemolytic anemia in the final project. Let's go check it out.
Like I said earlier, we're going to be exploring hemolytic anemia and the cellular basis, impact, and the future outlook of this disease. So, to introduce you to this disease, it's a premature destruction of red blood cells leading to a shortage of these vital cells in the body. Normally, red blood cells have a lifespan of about 120 days, but in hemolytic anemia, this lifespan can be drastically shortened to just days or weeks. This condition can be both inherited and acquired, and we'll be exploring both aspects today. So, moving on to the cellular basis, metabolic pathways, and enzymes damage. So, G6PD, it's a crucial enzyme in the pentose phosphate pathway. Note the stars by the G6PD, responsible for generating NADPH, which is essential for protecting red blood cells from oxidative damage. A deficiency in G6PD reduces the cell's ability to combat oxidative stress, leading to hemolysis. So similarly in pyruvate kinase, it's vital in the final step in glycolysis, seen here. Ooh, there you go, where ATP is generated. Mutations in the PKLR gene impair this enzyme, reducing ATP production and compromising red blood cell function and survival. So, in essence, to these key metabolic enzymes directly disrupts the normal functioning and lifespan of red blood cells. So, moving on, the functions of the cells impacted. The impact of hemolytic anemia extends to several critical functions within the red blood cell itself. So, first and foremost, oxygen delivery is impaired due to the reduced number of healthy red blood cells. So, energy metabolism is significantly affected, reduced ATP production, particularly in PKD, impedes the operation of ion pumps, membrane repair mechanisms, and this leads to the misshaped, dehydrated cells that are prone to rupture. In the G6PD deficiency, the impaired protection from oxidative damage accelerates cell destruction. So, as a direct consequence of these cellular dysfunctions, red blood cells become fragile and their lifespan dramatically shortens from, as we said before, 120 days to a few days or weeks. So, the consequences of hemolytic anemia are not confined to individual cells. They have widespread systemic effects on organs and organ systems. So in the cardiovascular system, the heart is forced to pump harder to compensate for reduced oxygen carrying capacity, which can lead to an enlarged heart. In the spleen and liver, which are responsible for clearing the damaged red blood cells, they can be enlarged due to being overworked, and this can lead you down the road of a splenectomy. When red blood cells are destroyed intravascularly, the released hemoglobin can overload the kidneys, causing renal damage, and furthermore, reduced oxygen to the brain can result in fatigue and some neurological symptoms. In autoimmune forms of hemolytic anemia, the immune system mistakenly attacks the red blood cells, exacerbating tissue damage throughout the body. The function of the body impacted. Beyond the organ level, hemolytic anemia impairs several key physiological functions of the body. Patients commonly experience fatigue and weakness due to decreased oxygen delivery, which limits physical activity and affects aerobic metabolism. The cardiovascular system responds to the oxygen shortage with an increased heart rate, and the liver and kidneys, crucial for the waste clearance, can be strained by the excessive breakdown of red blood cells. And in the autoimmune cases, immune dysregulation can lead to chronic inflammation, affecting the body's ability to distinguish self from non-self, furthering worsening anemia and related symptoms. Heritability and epigenetic influences. So, hemolytic anemia presents a complex picture regarding its origins, as it can be both inherited and acquired. Inherited forms such as hereditary spherocytosis, sickle cell anemia, and thalassemia are passed down through mutations in red blood cell membranes or hemoglobin genes. For example, in hereditary spherocytosis, it's often caused by mutations in genes like ANK1, affecting red blood cell shape and leading to early breakdown. In acquired forms like autoimmune hemolytic anemia, also known as AIHA, are not inherited but can be influenced by epigenetics. So, immune checkpoint inhibitors used in cancer therapy can cause epigenetic changes like altered DNA methylation in immune cells, leading to attacking the red blood cells. So, regarding inheritance patterns, hereditary spherocytosis is typically autosomal dominant, meaning a 50% chance of transmission from an affected parent. For sickle cell disease and thalassemia, they are both autosomal recessive, requiring both parents to be carriers for the child to be affected. And for G6PD deficiency, it's actually an X-linked, more common in males than females, and it affected more for males, but the females can be carriers if both X chromosomes carry the mutation. It's important to note that hemolytic anemia is not caused by failures in the cell cycle, and as mature red blood cells lack a nucleus and do not divide or undergo cell division. Moving on to genetic basis and mutations. For sickle cell disease, a mutation in the HBB gene, which encodes for the beta globin chain of hemoglobin, is responsible. Specifically, it's a point mutation where GAG changes to GTG, seen here, causing a glutamic acid to valine substitution, leading to the characteristic of the sickle cell shape. For G6PD deficiency, it's caused by mutations in the, no surprise, G6PD, often missense mutations, which result in reduced or dysfunctional enzyme activity and impair the cell's ability to manage oxidative stress. Pyruvate kinase deficiency involves mutations in the PKLR gene, including amino acid substitutions and reduced mRNA expression, leading to decreased pyruvate kinase activity and red cell fragility. And so these gene sequences are very well known, and genetic testing is recommended. Risk factors and lifestyle habits. So, beyond genetics, several lifestyle habits and other factors can increase the risk or trigger hemolytic episodes. So, for G6PD deficiency, certain foods like fava beans, medications like sulfa drugs, and environmental toxins like naphthalene can trigger red blood cell breakdown, unfortunately. Infections such as Epstein-Barr virus, EBV, can cause or worsen autoimmune hemolytic anemia. Additionally, autoimmune conditions like lupus and rheumatoid arthritis are linked to acquired forms of the disease. So, going on to diagnosis methods. Diagnosing hemolytic anemia often begins with routine blood work that reveals signs of red cell breakdown. So, initial indicators include decreased haptoglobin, an elevated reticulocyte count, and increased levels of lactate dehydrogenase and unconjugated bilirubin. So, a peripheral blood smear can provide crucial clues by showing specific abnormalities, such as in sickle cell, it would show sickle cells, or the bite cells that I showed earlier with those like little chomps in them, is what it looks like for the G6PD deficiency. Um, there are confirmatory tests, vary depending on the suspected type. So we have the enzyme activity assays for G6PD and pyruvate kinase deficiencies, and we have hemoglobin electrophoresis and high performance liquid chromatography for um, diagnosing sickle cell. In many regions, there is a newborn screening for sickle cell disease and G6PD deficiency for early identification. However, referral to a specialist, like often a hematologist, is typically needed for diagnosing these diseases. All right, moving on. So, available treatments and preventive technologies. While there's no way to prevent most inherited forms of hemolytic anemia, complications can be certainly reduced. So, for G6PD, avoiding oxidative stress triggers is key to preventing hemolytic anemia episodes. So that's going back to early detection through newborn screening is a vital um, thing that can help with proactive management for G6PD deficiency. For sickle cell disease, preventive measures, early diagnosis include penicillin, prophylaxis, vaccinations, and parental education to reduce early life infections. There's different treatments for specific types and severities of hemolytic anemia, but for like acute episodes, blood transfusion is a common option. For the autoimmune types, immunosuppressants like corticosteroids are used, and the intravenous immunoglobulin can help with the red blood cell destruction. Like I said earlier, a splenectomy may be considered for chronic unmanageable cases where like your spleen gets too enlarged. For sickle cell management, it includes hydroxyurea to stimulate fetal hemoglobin production and reduce sickling. And recently, there have been approved therapies like often additional symptom relief, offer additional symptom relief. For the PKD, treatments include folic acid supplementation, transfusions, and sometimes a splenectomy. There is actually a promising drug coming like now called mitapivat, which activates pyruvate kinase and has shown efficacy in improving hemoglobin levels and improved for certain patients. So the goal is to stabilize red blood cell and minimize symptoms and further prevent organ damage. So the outlook for management and active research, um, the outlook for managing hemolytic anemia is continuously improving, and thanks to advances in diagnostics and therapeutic options, there's a couple we can look at now. For G6PD deficiency, lifelong management often involves avoiding the triggers we mentioned earlier, but they're looking into research on mutation-specific diagnostics and antioxidant therapies, uh, ongoing research currently. But for sickle cell disease and PKD, novel therapies and gene-based approaches are revolutionizing care. So active research is underway in gene editing, like CRISPR, Cas9 for sickle cell disease, showing promising early outcomes in clinical trials. They're also exploring autologous stem cell therapies to reduce immune complication risk. And like I said earlier, in PKD, mitapivat is emerging as the first disease-specific therapy targeting underlying enzyme defects. So, holds a promising future for the individualizing the care of patients with different kinds of hemolytic anemias. All right, so the role of the allied health practitioners, they play a crucial role in the quality of life of these patients with hemolytic anemia. So dietitians help optimize nutrition by addressing deficiencies in iron, folate, and B12, and advising those dietary triggers such as flavavines in those G6PD deficiency patients. For dialysis, um, technologists, they manage the renal support for the patients that the hemolysis has caused kidney injury, and physical therapists assist patients in managing the fatigue that patients feel and coming up with a plan that includes like low impact exercises to help safely build endurance. So, in conclusion, hemolytic anemia is a diverse group of conditions characterized by premature destruction of red blood cells, impacting vital cellular function and leading to widespread systemic effects. We specifically covered sickle cell disease, pyruvate kinase deficiency, and glucose 6-phosphate dehydrogenase deficiency today. But there's a couple others that we weren't able to have enough time to explore, such as thalassemia, and the autoimmune condition that can come from hemolytic anemia as well. Unfortunately, while there is no universal cure, ongoing research and advancements in targeted therapies, including gene-based approaches, offer a promising look for improving patient care and their quality of life, which is the most important thing. Thank you for watching, listening, and these are my references. Goodbye.